So, Sana, can we just recall what we have done in the previous class?
Um, we did we talk about DNA replication?
No.
Oh,
We spoke about carbohydrates, we spoke about proteins.
oh right.
Okay. All right.
Yeah.
Okay. All right. So I just want to do few small things here. Last time we have done about carbohydrates, we have done about proteins. So I'll just show you this is what we have done last class. Okay. All right. So we did about the carbohydrates, we did polysaccharides, disaccharides. Okay.
Yeah.
Okay. So this was the amino acid. So today we are just going to do a little bit of the structural part. Okay, structural part. So let's see. First of all, and we're going to do something about the bonds also. We're going to see the different bonds. Okay, we have a glycosidic bond. We have peptide bond, right? Okay,
Yeah.
so all the different bonds are there. So a few of the bonds we are going to just try to understand them. Then we are going to go into the metabolism. Have you done metabolism?
Yeah.
Yes. Have you done metabolism? Yes.
Yeah.
Okay. So you must know what is metabolism, right? So okay. So we'll go through the metabolism and a few more things. So let's get started. Okay. So let's see here. All right. So when we talk about glucose, okay, when we say glucose, let's see the structure of glucose.
Mhm.
Okay. So you see a single, you can see a single oxygen atom. Can you see the single oxygen?
Yeah.
Yeah, it is attached to one, two, three, four. Okay, one, two, three, four. Here five, and here six. Six carbon atom. One oxygen is attached to one oxygen here. Within the ring, there are five carbon atom. Carbon
Mhm.
number one, carbon number two, three, four, and five. So when a single oxygen atom is attached to five carbon atom, we call this ring structure as pyranose ring structure. Okay, pyranose ring structure. Now the sixth carbon atom is outside the ring, so we're not considering that in the ring structure. Okay, so now what we are going to see is how the other molecules, because glucose is a compound of carbon, hydrogen, and oxygen. Okay, so let us see here. So we have a hydrogen here, OH. Then we have a hydrogen and another OH, and then we have an OH, and then we have a H. So let's see here. Okay, all right. So this would be the structure of glucose. Now, what we need to understand here is that glucose is a carbohydrate. We have the second carbohydrate, like for example, it can be fructose, it can be galactose. Now, we all know that one carbohydrate will join with the other carbohydrate to form either oligosaccharide or polysaccharide. And this bond which connects them, this is called as what bond? It is called as glycosidic bond. Clear? It is called as what? Glycosidic bond. Clear?
Yeah.
Okay. One second. Uh, one second. Okay. All right. So glycosidic bond. All right. So we are going to see now how this glycosidic bond will be made. Okay. Now glycosidic bond are made by the removal of a water molecule. In the book also, you have this particular section and you need to know this. Okay. Have you studied this, Sana?
Yeah.
You have studied this. So, right, it'll be easy for you. Okay. So let us see the bond between glucose and glucose. So we'll keep it clean. So let's see here. Okay, we have one glucose molecule here. Then we have an H, OH. Then we have a H, OH here. OH, clear? Okay. Now we have this. So this is carbon number 1. Okay. So we know the numbers. Carbon number 1, carbon number 2. This is 2. This is 3. This is 4, 5, and 6. Okay. So now how between this is one glucose. This is glucose molecule, and we will see another glucose molecule. Okay. Now let's see here. Okay, so we have another glucose molecule. Okay, all right. So the same thing will be drawn here. Okay, we have H, OH, OH, and OH is hydroxyl group, and then we have an H, OH here, and then this will be H and CH2OH. So this is another glucose molecule. This is another glucose molecule. So now we are going to join. So glucose plus glucose will join to form maltose. Okay, will join to form what? Maltose. Clear?
Yeah.
Okay, glucose plus glucose will join to form what? Maltose. So let us see here. Now, this they will join together by what we call as a glycosidic bond. Okay, so glycosidic bond will be formed by elimination of a water molecule. So let's see here. Can you see this red line I am drawing? Look very carefully. So H2O. So this is one removal of H2O. Okay, so when the water is removed, okay, I'll show you. This is what we get. Okay, when we remove the water from this region, okay, when we remove the water, we will get the oxygen left. Okay, can you see this, Sana?
Yeah.
This is glycosidic bond. See, this is the
Yeah.
bond. This for every carbohydrate, this is what it will form. Now, tell me one thing. Is this carbon number 1?
Yeah.
Yes. And what about this?
No.
Carbon
That's
number, see, count. This is 1, this is 2, this is 3, and this is what?
4.
4. So the bond is formed between carbon number 1 and carbon number
4.
4. Okay. So, since it is on the same orientation, we call it as the alpha 1 to 4 glycosidic bond or linkage. This point is clear, Sana?
Yeah.
Glycosidic bond or linkage. Alpha 1 to 4 glycosidic bond or
Okay.
linkage. Clear?
Yeah.
Now, next, see here. Let's see. Okay, so this is one. Now, suppose, for example, okay, suppose, for example, we have another glucose molecule. Asana, are you understanding?
Yeah.
Okay. Now, see, suppose we have this another glue. Let's not see glucose molecule, not say glucose. Let me think, let me think. Ha, okay. Lactose we will take. So, glucose plus galactose. Okay. Now, so H, OH, H, OH, hydroxyl, water, H, H, CH2OH. Okay.
Mm-hmm.
Okay. So this is galactose. This is galactose. Let me draw the same glucose molecule here again. Okay. So this you'll be knowing the same glucose molecule OH, H, then we will have H, OH, let's see, OH, H, okay, and then we have H, OH, and of course this is the sixth carbon CH2 OH. Now look into this very nicely. The orientation is very different. This is elimination of one water molecule. So one water molecule is eliminated. So how will you get the bond? I'll draw in blue. So you will get the bond like this. Clear?
Yeah.
Did you get that in purple? Yes.
Yeah.
This bond is called as beta 1 to 4 glycosidic bond or linkage. Clear?
Mm-hmm.
Okay. A third one. A third one. Let's see. We'll take glucose. Sana, are you understanding?
Yeah.
Okay. Have you done this before?
Yeah.
All of it?
Yes.
Okay. So it's easy for you to understand, right?
Yeah.
Okay. So we draw a glucose here. Okay. So we take a glucose here. CH2OH. Okay.
Mm-hmm.
All right. Let me take the other orientation. Laboratory. Okay. Yeah. Perfect. No problem. HOH. Okay. All right. This is a glucose. The next that I'm going to draw is a fructose. Okay. So fructose. Let's see. CH2OH. This is OH. Then here we have an HOH. This would be OH. H. And then we have HCH2OH. This is a fructose, and fructose is a fruit sugar. Okay. Now, in fructose, we count it in different way. This is carbon number 1. This is carbon number 2. This is 3. This is 4. This is 5. And this is 6.
Do you only count it like this in fructose?
Yes, if this is what we do it in fructose. Clear?
Okay.
Yes, fructose and glucose are isomers.
Mm-hmm.
Okay, same number of carbon atoms, but different structures.
Yeah.
Clear?
Yeah.
Okay, this you need to know. All right. Okay. Now, Sana, tell me what bond will it be? I'm eliminating a water molecule.
Is
Yes.
glyco
Yeah, glycosidic.
glycoside.
Alpha. Is it 1 to 4 or 1 to 2?
1 to 2.
Yes. This is 1 to 2. Glycosidic linkage or bond. This point is clear, Sana?
Yeah.
See, understood or not? Yes?
Yeah.
Okay. Now, next, let us see here. 1 minute. Okay. Let us try to understand this. Okay. Now, since you are well aware of the what do you call that? The carbohydrates, glycosidic linkage. Let us throw some light on the polypeptide linkage or the peptide bond. So, Sana, we know that peptide bond is formed between two amino acids, right?
Yeah.
Okay. Now, suppose, let's see, we have an amino acid. This is the alpha carbon atom. Okay, we have H. Sorry, this is N, NH2, NH2C. This is R, and there would be C double O and OH, carboxylic group. Okay, plus C, same thing we are going to write again. The same thing we are going to write again. Okay,
Yeah.
all right. Now, in the last class, I mentioned one thing. The amino acid will have an N terminal, and amino acid will have a C terminal, right?
Yeah.
So, Sana, can you tell me which is the C terminal and which is the N terminal?
Um. The
Which is C terminal? Yes,
the
region. Can you see
what?
this?
Yeah.
Yeah, this is the carboxylic acid group is attached. So, this is the C terminal,
Okay.
and this is the amino group. So, this is the N terminal. Clear?
Okay.
Now, here also there will be elimination of water molecule. So this water molecule is eliminated. This water molecule is eliminated. Okay, the water after the elimination of the water molecule, after that elimination of the water molecule, this is what we will get.
Mhm.
Let's see, N or NH2 you can write, no problem. H R C double bond O N H, because we have removed one water molecule, right?
Yeah.
Yeah, we have removed one water molecule. Yes. 1 second. Mm. I forgot something to write here. Sorry. H and this is R. That's what I was wondering. Okay. All right. So, okay. Let me erase this. Mm, H. H. Oh. Sorry. Okay. H, R, and C, O, H. So, we have a glycosidic bond. Okay, sorry, we'll have a peptide bond. So this here, this is the peptide bond. Clear, Sana? Yes, understood?
Yeah.
Yes. So this is how the bond formation takes place. Okay. The next thing that we need to, we have studied about the protein structure, the primary, secondary, and tertiary structure, right? Yes.
Yeah.
So we'll do one thing, we'll throw some light on the properties and the functions of proteins. Okay, I'm going very stepwise, Sana, so that you understand the things very properly. First, it's always important, because these sections come under molecular biology. So whenever you study molecular biology, you should start with the basic first. You understand the basic, you are able to understand the entire molecular biology very nicely.
Okay.
So just like a plant, if the plant's root is not proper, will the plant be able to grow?
No.
No. Okay, so you need to keep your basics strong. So that's what I'm doing. Okay, I'm doing the basics, and then important ones, it's very stepwise manner.
Yeah.
Okay. Yeah, so I'll do everything, you don't worry. You don't think that, okay, this was there, ma'am did not do. No, ma'am is going to do everything in a very sequential manner. Clear?
Okay.
Mm. Now let's see some functions of protein. Protein functions. Okay, this is important. Protein functions, how protein will function. Okay, now, if whenever we carry out any reaction, reactions are catalyzed by what? Enzymes.
Mm-hmm.
Okay, enzymes control the rate of reaction. Enzymes are protein in nature. We are going to study that. Enzymes are protein in nature. Now, in our body, we have muscles. See, when I am writing, the muscles of my hand are contracting. Okay, muscles of my hand are contracting. So, what is happening here is that when muscles are contracting, in muscles, we have proteins like myosin, we have actin, we have troponin, and we have tropomyosin. Okay, troponin, tropomyosin. Clear? All right. So now try to understand this. In muscles, all these proteins which I named, these will undergo contraction. It'll undergo contraction if it has to show any kind of movement to the muscles. So muscles contain protein, which when they will contract, the muscles will perform their work.
Okay.
Next is cytoskeleton. If this is our cell, this is our nucleus. Inside the nucleus, we have cytoplasm. Cytoplasm.
Mm-hmm.
Okay, cytoplasm will contain a type of supporting skeletal network which is called as cytoskeleton. The cytoskeleton supports the organelles, and it will also act as a supporting skeletal network or framework for the cells. So cytoskeleton contains microtubules. Microtubules are protein, and they are made up of protein called as tubulin protein.
Yeah.
Okay, tubulin protein. So these tubulin protein, all right, they will help in the cyclosis or movement of the cytoplasm. Okay, movement of the cytoplasm. Tendons, ligaments, okay, all of these are maintained by uh, different proteins. Okay, our tendons, our ligaments, they get tensile strength. Okay, so that comes from protein. So we call protein as the building block. Okay, protein is called as the building block of the body. It is important for maintaining the various body structures. Okay, it's important for maintaining the various body structures. Now, blood coagulation, that is blood clotting. Blood coagulation or blood clotting will also carry out by various factors which are protein in nature. The exchange of gases, okay, exchange of gases, all right, exchange of gases will also take place, or exchange and transport transport of gases takes place by a protein which is called as hemoglobin. Okay, exchange of gases and transport of gases from alveoli to different parts of the body, that is with the help of hemoglobin. And hemoglobin is protein in nature. Okay, protein in nature. Cell adhesion, membrane transport of membrane in cell. Okay, we'll see a membrane transport. That means how the components are transported from one cell to the other.
Yeah.
So membrane transport. Okay, membrane transport. All right, then when we talk about membrane transport in the cell membrane, we also have the transmembrane protein. The transmembrane protein in the cell membrane or plasma membrane is also protein in nature.
Mm-hmm.
And the transmembrane protein in plasma membrane helps to transport water soluble substances across the cell. Okay, water soluble substances across the cell. The hormones that are produced in the body, hormones that are produced in the body are also protein in nature. Hormones are chemicals secreted in the body. They they function in very different manner. Okay, hormones, cell receptors. Then the packaging of dna packaging of dna
Mm-hmm.
in our chromosome is due to the presence of a protein. Okay, due to presence of a protein which is called as histone protein. Okay, histone protein. That very important. The structures that provide immunity to our body, the antibodies. Okay, the antibodies are also protein in nature.
Mm-hmm.
Okay, antibodies are also protein in nature. These are some of the most important functions of the protein. Okay, some important functions of the protein. Is it clear, Sana?
Yeah.
Okay, now we have done about protein. Okay, in the next class we will slowly go to the lipids. But before that, since we have done about protein, we need to study about the water in the body. We need to study about the metabolism, the anabolism, and the catabolism. Okay, so these points are clear, Sana. Any doubt here?
No.
Okay, all right. Now, see, whenever in our diet, suppose we are eating food, okay, and this food will be digested by our digestive system. Okay, it's digested by our digestive system. Okay, now listen to this very nicely. Okay, listen to this very nicely. All right.
Yeah.
So what happens? Suppose the food. Okay, so we have to digest. Now, suppose for example, we eat protein in our food, then protein has to be broken down. Once the protein breaks down from the food, the protein will start, okay, we can say denaturation of protein, or protein are being utilized. But when the protein is being broken down, okay, what do they release? They release some kind of waste. They release some kind of what? Waste. Okay, so let's see. Whenever there is metabolic reaction, we will always produce waste. These are not used by the body. Then, after the body has used the hormone, after the body has used the hormone, this will be eliminated in the form of what? In the form of waste. Okay, in the form of waste. That means the spent hormones, I can say the spent hormones that has been utilized. So that's the waste. Okay, then for example, if we are taking our metabolism will also generate carbon dioxide. Okay, our metabolic waste will generate gaseous waste, which is carbon dioxide, and the solute waste, which is solid waste, which is nitrogen. Sorry, see, gaseous waste is carbon dioxide, and solid waste will be nitrogenous compound.
Yeah.
Okay, nitrogenous compound. So now you see here, when food when our, when we digest food, we utilize the oxygen, we release carbon dioxide. Okay, we release carbon dioxide. So what happens? Carbon dioxide is a waste and needs to be eliminated. Okay, similarly, when hormones are utilized, we call them as spent hormones. Okay, spent hormones. So let us see here what is the fate of the hormone protein that we eat in our diet. Consider it the hormones, spent hormones. Consider the food which is rich in protein. Okay, food which is rich in protein. So whenever we are having lot of protein in our body, okay, we have lot of protein in our body. So what happens, that protein, some of them will remain as a waste material. Okay, some of them will remain as a waste material, and this waste material of protein we need to eliminate from our body. Remember this, we cannot allow proteins to accumulate in our body. Okay, we have to remove all the proteinaceous waste. So some of the proteinaceous waste, ammonia, are too large and cannot, and need lot of water, and we cannot remove them. So what happens? The nitrogenous waste will go to the liver. In the liver will perform ornithin cycle. Liver will perform what? Ornithin cycle. Is it clear? Yes.
Yeah.
Okay, liver will perform ornithine cycle. In ornithine cycle, what happens? Whatever ammonia, whatever the amino acids, extra amino acids, whatever extra protein which has entered the liver, they will be converted into, okay, they will be yes, these will be converted into, all right, the urea, which is the simple form that can be eliminated. So all the proteins, all the amino acids in our body cannot be at once eliminated. They all collect in liver. In the liver we carry out deamination. After that, in deamination, we'll have the ornithine cycle, where all the extra protein, extra nitrogen, everything is converted into urea. And it is urea which is eliminated in urine. Okay, urea will be eliminated in the urine. This point is clear, Sana?
Yeah.
Yes, any doubt here?
No.
See properly. All clear? Yes?
Yeah.
Okay. So, in our body, because of metabolism, we produce a lot of waste. That waste goes to the liver and then finally it is eliminated along with urine. So, we have the Ornithine cycle. We have the Ornithine cycle where ammonia combines with carbon dioxide to form ammonium ammonium carbamate.
Yeah.
Okay, ammonium carbamate plus urea plus water. Okay, urea plus water. All right, so this would be the reaction pattern. Okay, reaction pattern. How in the liver we produce urea. Urea is safe to excrete. It requires less water for its excretion. And it is in this form that it will be easily eliminated from the body. Okay, till here, is it clear?
Yeah.
Yes, any doubt?
No.
Okay, all right. Then let's see. Okay, so when we talk about metabolism, Sana, can you define me metabolism?
The chemical reaction
Yes.
in which a living organism
Hmm.
maintains life.
Okay, and anything else?
Uh, yes.
Yes, that's it.
Uh,
Metabolism. How will you define metabolism? It is the sum total of all chemical reactions. Sum total of all chemical reactions occurring in our body is called as metabolism. Okay, it is the sum total of all the chemical reactions that is occurring in our body. So that's metabolism. So metabolism, let me write here. Metabolism is of two types. Okay, they've divided metabolism into two categories. One we call as the anabolism, another is the catabolism. Okay, one is what? Anabolism, and another one is what? Catabolism. Clear?
yeah.
So metabolism we know. Now what is this anabolism and catabolism? Sana, can you explain me this in short?
Anabolism is like um, when living
Yes.
organisms,
Hmm.
they like the synthesis
Okay.
of living organisms into simpler
Hmm.
ones.
Hmm.
And then catabolism is like the breakdown of complex molecules.
Huh. Complex molecules. Anabolism is called as constructive constructive metabolism. That means we are making complex compounds in anabolism from simple compounds. Okay,
Yeah.
write that down. Very important. In anabolism, we are making complex compounds from where? From the simple compounds. Clear or not?
Yeah.
Okay, from the simple compounds. So, for example, photosynthesis. Photosynthesis is that procedure with the help of chloroplast, pigments, water, minerals, light. The plant will make the complex compound called as starch. Okay, plant will make the complex compound which is called as starch. So by combining many simple forms, we have made a very complex structure. This complex is a polysaccharide. Okay, is this point clear?
Yeah.
Okay, next is catabolism. This is called as destructive metabolism. Destructive metabolism. So, from the word itself, you can make out the complex compounds in catabolism are broken down into smaller pieces, smaller simple pieces. Okay, suppose, for example, the person we take in starch. We will take in starch. Starch is a complex carbohydrate which is made up of many glucose units joined by glycosidic bond.
Mm-hmm.
Okay, so what happens here is that in anabolism the plants have made the starch. Now when we eat the vegetable we get the starch. Okay, so
Yeah.
now the starch, which is nothing but a polysaccharide, very important. Okay, polysaccharide, this will break down into simple components. Okay, because of digestion and digestive juices, the complex starch will break down into simple simple compounds that can be simple compounds that can be utilized. For example, starch is converted back to glucose. Okay, starch is converted back to glucose. So this is catabolism. Okay, catabolism. All right, so anabolism and catabolism. Is it clear?
Yeah.
Yes, anabolism and catabolism both are clear or not?
Yeah.
Okay, all right. So now when we talk about metabolism in our body, in every organ, in every cell, okay, metabolism is being carried out. Okay, metabolism is being carried out. So here what we need to understand, is here, like for example, let us see here. Okay, so since every cell has metabolism, okay, since every cell is having metabolism, so it can be either anabolism or it can be catabolism.
Yeah.
Okay, right? Yes. So how do this metabolism occur? The steps of metabolism are right here. The steps of metabolism is called as metabolic pathway. Metabolic pathway. The steps of metabolism is called as metabolic pathway. Now every step every step is, okay, every step will be under the control of enzymes. See the relation,
Yeah.
right? Every metabolic step is carried out in various step. Okay, we call that pathway, and every step has to be controlled by whom? Enzymes. We may have linear metabolic pathway. For example, glycolysis. In short, if I say what is glycolysis, this is the metabolism of glucose is called as glycolysis.
Mm-hmm.
Okay, linear pathway. We also have circular metabolic pathway. That means it occurs in circle. So we see this in the process of photosynthesis, and also in the respiration. Photosynthesis, Kelvin cycle, which is the dark reaction. Respiration is also Krebs cycle. Okay, Krebs cycle. So metabolic pathway can either be linear or it can be circular. Is this point clear?
Yeah.
Okay. Now, when when we are talking about metabolism, it is important for us to understand few important things which is related to what we call as enzymes. Okay, enzymes. We need to study the enzymes now. Okay, can you define me what is an enzyme?
Um, it's
Yes.
um, it's a substance
Hmm.
that acts like a catalyst.
Yes. Okay, that acts like a catalyst. Yes or no?
Yeah.
Okay. All right. So now let's see here this fact. Okay. All right. So we have lot of enzymes. Okay, we have lot of enzymes, and and we have lot of metabolic reaction. So please remember, every step of every metabolic reaction is enzyme specific. It's
Mhm.
not like same enzyme will be carried out for so many steps or will be used for so many metabolic steps. No, one enzyme for one step. It can be one enzyme for two step also, but not more than that. All right, so enzymes are very specific. Okay, now what we will do is that, okay, so now we'll go on sequentially. So now we are going to study about the enzymes. We have studied about the proteins, we have studied about the bonds in the proteins. Now we will study the enzymes. Okay, is it clear till here? You have any doubts, Sana, you can ask before I go into the enzyme section.
I understand.
Yes, all here?
Yeah.
Okay, let's see. Okay, enzymes. All right. Okay. Okay. Before that, so now we'll do few things here. Let me just one second. Okay. Now let me bring it here. Okay. So before we do enzymes, we need to study about some peculiar things. Okay. So now you have your textbook with you.
Yeah.
Yes. Can you take out page number 93? Can you take up page number 93? Are you on that page?
Um, yeah.
Okay. Can you see the second activity down? Activity. Can you see something written as Botox? Can you see page number 93? Are you on which same book you're using, right?
Yeah.
Hmm.
Oh, yeah, I see it.
Yes. Okay. Can you see Botox?
Yeah.
Yes. Now, see what it is written. Botox. It's a
Neurotoxin.
neurotoxin. Another protein. Okay, neurotoxin, another protein. All right, so neurotoxin means it'll affect the nervous system. Okay, so let's see. Botox is obtained from, it is written as Clostridium. Clostridium botulinum. Okay, Clostridium botulinum is a bacteria, and this bacteria is responsible for botulism. Okay, it's responsible for botulism. Can you tell me what is botulism commonly called as?
Um,
Yes,
I'm not sure.
food poisoning.
Oh,
Okay,
mhm.
1 second. Okay, food poisoning. Okay, food poisoning, botulism or food poisoning. Now what happens, this botox, okay, this botox is also used for some, let's see what it is used. Okay, this botox is a neurotoxin, and it finds its uses. Okay, so let's see here. Botox will be extracted from the bacteria. Okay, and in very small doses. Okay, very small doses. What happens here? It will be used to reduce. It will be used to reduce wrinkles. Okay, it'll be used to reduce what? Wrinkles. Okay, so all right, wrinkles, you understand, Sana?
Yeah.
Yes, it will be useful to reduce wrinkles. So in surgery, and they do the skin upliftment and all. Okay, so they use this Botox in very small amount. Okay, very small amount, so that it cannot have any side effects. Clear?
Yeah.
Okay. Don't forget botulinum. Okay, Clostridium botulism. Is this clear?
Yeah.
Okay. All right. So let's see some of the common examples of protein, okay, which is given here. All right. So first we will come to Rubisco. Let's see here. Rubisco. Okay. So Rubisco is the most abundant protein. Okay, in the world. All right, Rubisco, its name, Ribulus, Ribulus,
Yeah.
bisphosphate. C stands for carboxylase, and O stands for, okay, Rubisco bisphosphate carboxylase, oxygenase. Okay, oxygenase. Rubisco. Rubisco is that, okay, that protein. It can be called as an enzyme also. It is rightly called as protein. Rubisco is that protein where the Rubisco, okay, Rubisco is utilized, or is, it will carry out dark reaction. Okay, dark reaction of what? Dark reaction of photosynthesis. Dark reaction of photosynthesis. Okay, and what does Rubisco do? Rubisco is able to trap carbon dioxide and carry out with the process of what? Photosynthesis.
Yeah.
Now, why carboxylase? Whenever the carboxylase part, okay, whenever the carboxylase is activated, that means it will carry out fixation of carbon dioxide. And whenever we see oxygenase, suppose for example, okay, if in a plant there is more carbon dioxide, less oxygen. More carbon dioxide means Rubisco will fix all carbon dioxide and take them for photosynthesis.
Okay.
Now what happens? The carboxylase part is not there. It's only oxygenase. So oxygenase, okay, will what it will do? See, suppose for example, carboxylase will trap CO2 when the CO2 is more. Oxygenase, what it will do? It will trap, one second, oxygenase will trap oxygen and it will obstruct the dark reaction. Oxygenase will trap oxygen and disrupt the dark reaction. Clear on this?
Yeah.
Okay, Rubisco, carboxylase, and oxygenase. So when you study photosynthesis, any compound which is what it is doing is that when it is fixing carbon dioxide, it will be the carboxylase of Rubisco, and when it is fixing oxygenase, okay, when it is fixing oxygenase, it is if see, carboxylase oxygen, oxygenase means it will trap what? Oxygen instead of carbon dioxide. Clear on this?
Yeah.
Yes, any doubt here?
No.
Mm. Next, insulin. Yes, Sana, tell me about insulin.
Um, it's like it's produced in the pancreas.
Yes, it is produced in where? In the pancreas. Yes or no?
Yeah.
Yes. So this is produced by the beta cells of pancreas. Beta cells of pancreas. One second. Beta cells of pancreas. Okay. Now, let's see here. Insulin regulates the glucose level. Okay, it will decrease the blood blood glucose level. Insulin decreases the blood glucose level. Insulin is also a protein. It has two polypeptide chain. One is alpha chain, okay, and another one is beta chain. Insulin have, okay, so it has alpha chain and beta chain. All right,
Okay.
is this clear? Yes.
Yeah.
Insulin's function is to remove the glucose. Insulin is also a protein. Next protein that we will see is immunoglobulin or Ig, immunoglobulin. So we have different type of immunoglobulins. Okay, so how will you remember them? Let's see here. First of all, what are immunoglobulins? Immunoglobulins are nothing but they are proteins that will produce antibodies in our body, and antibodies will help us to fight against infection. Okay, it will help us to fight against what? Infection. Is this clear?
Yeah.
One second. Okay, all right, immunoglobulin. So we have IgG, immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin E, and immunoglobulin D. Okay, how do you pronounce this?
Um, ramen.
Gamed, right? GAMED. Yes or no?
Yeah.
Yes. Okay, see? GAMED, gamed. You just have to remember this word gamed. And immediately you can just add immunoglobulin G, immunoglobulin A, M, E, and D. Clear?
Yeah.
Okay. So let's see here. Okay, all each of this play very specific roles. Okay, then I last week I had mentioned about collagen protein. Collagen protein are the fibrous protein which is present in our skin and helps to formation of new skin. Clear?
Yeah.
Rhodopsin. 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 Rhodopsin is the visual pigment. Okay, this pigment will help us to see. Okay, rhodopsin. All right. Now, when we talk about rhodopsin, okay, rhodopsin in the bright light will break up into its components called opsin, opsin and retinal. Retinal is a constituent of vitamin A.
Mhm.
Okay, so rhodopsin in the presence of light. Okay, when you're going in bright light, rhodopsin will break into two components, opsin and retinal. Okay, opsin and retinal, and this opsin and retinal will help you to see in the very bright light. Clear?
Yeah.
Okay, rhodopsin. All right. Then spider silk, and we have different type of silk that is secreted by the silkworm. Okay, silk moth or silkworm. So all these silk, okay, silk whether secreted by the fiber, secreted by the silk moth. All right. So these all are what they are silk. Okay, they are silk, and they have special property. The spider silk is very tough. Even the silk that comes, the silk that comes from, like for example, like insects, silkworm, that is also what happened here, silkworm. All right, that is also very tough. So silk will contain a protein which is called as fibroin. Fibroin is the protein of silk. This is the protein of silk. This point is clear?
Yeah.
Yes. Okay. So till here, any doubts, Sana?
No.
No? Sure?
Yeah.
Yes. Can I
Yeah.
ask you questions then?
Sure.
Okay, fine. I'll ask you at the end. Okay? Okay. Before that, let's see here. Okay. Okay. Now, let's see. We talk about, let's see, enzymes. Okay, enzymes. So enzymes are catalyst. These enzymes are what they are, catalyst, and they accelerate the rate of reaction. Okay, enzymes are catalyst. Okay, they are organic catalyst, and they accelerate the rate of a chemical reaction. Accelerate means that these, okay, these what it will do, it will make the reaction go more faster. Okay, it'll make the reaction go more faster.
Yeah.
Now, few things you have to understand. See, Sana, here, very important thing. Okay, when, suppose for example, you are carrying out a chemical reaction, you want to start the chemical reaction. So when you have to start the chemical reaction, you have to give the initial heat to the chemical reaction. Okay, when you are starting a chemical reaction, you have to provide a heat to the chemical reaction. So that heat is called as activation. Activation energy. Any chemical reaction, when it starts, there should be a pushing energy, initial energy that will start the reaction. And that energy is called as activation energy. This point is clear?
Yeah.
Okay. Activation energy, okay, sometimes activation energy are very high, and when it occurs in very high activation energy, so the production of products will be what, affected. Okay, products will be affected.
Mhm.
So what do we do is that we use an enzyme. We use an enzyme in any kind of reaction. The enzyme will reduce reduce the activation reduce the activation energy. Okay, enzyme reduces the activation energy. Now that means now the enzyme, now the reaction will be very faster. Not only faster, but energy, the lot of heat production also will not be there. So enzyme reduces the activation energy. Okay, is this clear?
Yeah.
Okay, it reduces the activation energy. Okay, let's see. Now enzymes are specific. Enzymes are specific. As I said, that every metabolic reaction, okay, every step of metabolic reaction is carried out by separate enzymes. Okay, so enzymes are very specific. Keep this in mind. Okay,
Mm-hmm.
next. Okay, next. Yes, enzymes. Okay, now what else can I say? Can you tell me some more points, Sana? Yes,
Um,
two
they
points I have told you. Now you tell me the others.
um
Yes, think.
um pH
Sorry,
pH levels.
pH that we will study in the factor.
Okay.
We will study in the factor. Okay, now you see enzymes reduce activation energy. Enzymes are specific. Okay, so when any reaction contains enzyme, okay, when any reaction will have enzyme, the production of products will also be increased. Is this clear?
Yeah.
Okay, production of products will also be increased. Okay, so let's see here. Okay, so in your book we have the factors. All right, so let's see about an enzyme now. Okay, let us see the various feature of an enzyme. So I've written here, enzymes reduce the activation energy, and enzymes are what? Specific. Okay, specific, but I've not told you the drawback of enzymes. We'll come to that drawback later on. Okay, we'll come to that drawback later on. Clear?
Yeah.
Okay. All right. Okay, fine. Let's okay. So let's see here. One second. Okay. Now, Sana, tell me, you know the structure of enzymes?
Yes.
If you remember, I'll give you 1 minute. Just 1 second. Okay. Now, do you remember in the last class we had done about protein conformation, protein structure, primary, tertiary, secondary structure of enzymes, of protein,
Yeah.
sorry.
Yeah. Yeah.
If you, yes. Do you remember this? Do you remember this? See?
Oh yeah.
Yes. Can you see this?
Yeah.
Yes. Yes. So enzymes are nothing. Okay, enzymes are nothing but what are enzymes? Enzymes are protein. Okay, enzymes. Enzymes are what? Enzymes are protein in nature. Keep this in mind. Now, listen to this very nicely. I have an enzyme with me. Okay. Now, an enzyme will have what? Enzyme, if you remember, I said that enzymes will have a specific site which is called as active site. Okay, enzymes have a specific site which is called as what site? Active site. Now, how will the enzyme function? First, I'll show you the structure, then we will go to the enzyme function. Okay, all right. Okay, there we have a beautiful red color enzyme, right?
Yeah.
Okay, now this is okay, this is called as Apoenzyme. What is it called? Apoenzyme. Because this enzyme is only the protein part of the enzyme. It is only the protein part of the enzyme. So it's called as what? Apoenzyme. Now, this enzyme alone cannot function. Alone cannot function. It should
Yeah.
always have, okay, it should always have a cofactor. Enzyme should always have what? Cofactor. And the cofactor will be attached to the enzyme like this. So this is what is this? This is a cofactor. Okay, so this is the cofactor. Now, once the cofactor is attached to the enzyme, okay, even the substrate will come and attach to the enzyme, and enzyme will become active. Are you understanding, Sana?
Yeah.
Yes. So this is the substrate. The enzyme have to change this substrate. Clear? Enzyme have to change this substrate. Now this entire enzyme is an active enzyme. So this is an entire active enzyme. Entire active enzyme, which is called as hollow enzyme. Clear?
Yeah.
Yes. So can you explain me the structure?
Yeah, so
Hmm.
okay, so there's the active site,
Hmm.
and the active site is made of proteins.
In the entire enzyme is made up of protein.
Yeah,
Active
the
site
entire.
also. Ah. Okay, then.
And then connected to the enzyme, there's the substrate.
Yes.
And
Because we have to change the substrate into what? Product, right?
yeah.
Yes. Okay. Next.
And then there's also the
Cofactor.
the cofactor.
Yes. So can a function, can a enzyme alone can function without a cofactor?
No.
No. So what is this total activated enzyme called as?
Um, it's called the hollow
See,
enzyme.
it is written down. Hollow enzyme. Very good. Okay. It's called as hollow enzyme. Now our work will be to understand what is cofactor then. Okay. What is cofactor? What are the different type of cofactor? So let's see the cofactors. We have three different type of cofactors. Okay. All right. We have sorry. We have apoenzyme. Next we have is coenzyme. Third one we have metal ion. Apoenzyme, okay, if attached to apoenzyme is organic part, and it is tightly bound to enzyme. Coenzyme is also organic part, but it is loosely bound to enzyme. And metal ions can be any metal ions, like for example Mg2 plus magnesium, clear?
Yeah.
Mn2 minus manganese, all right, anything, potassium, anything it can be. All right. Okay, so these are the components of enzymes. Did you
Yeah.
understand this, Sana?
Yeah.
Okay, so these are the components of what? Enzymes. Now what does enzyme do? Enzyme action. Okay, so let's see here. This is our enzyme. Okay, this is our enzyme. Attached to enzyme. Okay, and what happened? We have a small substrate. This is the substrate. This is the enzyme. And this is the substrate. Now it is this substrate that we want to change into product. We want that product. Okay, so next what happens is, okay, so this is the enzyme. All right, the substrate comes and binds to the enzyme at the active site and forms a complex which is called as enzyme substrate complex. Enzyme substrate complex. Clear?
Yeah.
Yeah. Enzyme substrate. When the enzyme, see yes, Anna, when the enzyme holds the substrate, it brings about changes in the substrate. And suppose my hand is enzyme, this pen is a substrate. So enzyme will hold the substrate like this. It will break the bond in the substrate. It will make new bonds. It will totally change the substrate. Okay. And when it is changing, there will be intermediate product may be formed. That means not the real product, but intermediate product means that there is a reaction going on. This stage is called as the transition stage. Clear?
Yeah.
Yeah. So transition stage will ultimately lead to, okay, lead to the formation of product. Okay, now the product will be formed. So I'll leave the product in another color. So this is the product that is formed. Instead of that, let me give a yellow color. That will be bright. Yes, a product is formed. Can you see this?
Yeah.
Yes.
Yeah.
So now this was substrate. Now this became a product. And this is called as enzyme product complex. Okay. Now, can I use this product, Sana?
Um. No.
No. So what I have to do?
Uh. Oh.
What I have to do?
You have
I have to
to.
separate, right, the enzyme and the product. Yes or no?
Yeah.
Yes. So now I will separate the enzyme and the product. So once separated, I get my product, and I have I have a free enzyme.
Mm-hmm.
What enzyme it is? Free enzyme. What enzyme?
Free enzyme.
Yes. Free enzyme. Free enzyme. Now this free enzyme will be, it will be, taken to combine to another substrate for the reaction to continue, and this product is extracted, it is removed, so that I get the product. This is nothing but what it is called as mechanism of hormone action. Mechanism of hormone action. Okay, mechanism of what action? Hormone action. Okay, now you tell me, Sana, suppose for example, let us see this. Okay, suppose if a reaction is there. Okay, you're carrying out a reaction. Okay, so we have many enzymes here, right?
Yeah.
Yes. We have many enzymes. So many enzymes are there. Now, each of this enzyme is occupied by a substrate. Each of them has a substrate. Clear?
Yeah.
Each of them has a bonded substrate. See, each of them will have a bonded substrate. Okay. Now, so after binding with the substrate, it is going to produce what? Product, right?
Yeah.
Okay. Now listen to this. When initially you had only the enzyme, okay, and you added the substrate, okay, first you had the enzyme, which is free enzyme. And what you added? You added the substrate. Can you tell me the nature of the reaction, Sana?
Um, it's
Can you tell me the nature of the reaction? What will happen? Will it be faster? Will it be slower?
faster.
Faster. The reaction will be very fast. Okay, reaction will be very fast. So, reaction, it will have a very fast reaction. All right, so you can see first initially, then you see very fast reaction, but after some times it becomes stationary like this. Can you tell me why? So we have a fast reaction here. Later on it becomes stationary. Can you tell me why?
Um, because that's
Yes, because why it becomes stationary?
um,
Here the reaction is fast, right?
yeah,
Yes, the reaction is fast because every enzyme is binding to the substrate. But
yeah.
here, why this is becoming stationary? Because at this point, all enzymes are saturated. Okay.
Mm-hmm.
See, all enzymes will be saturated. That means by the time you add, all enzymes are occupied. Every enzyme is occupied with a substrate and it's carrying out a reaction. Then at this point, you'll see this curve is there right here. Another point you will observe is this flattened line, which is called as what? Stationary phase.
Mm-hmm.
Okay, what phase? Stationary
Stationary.
phase. This curve is when all enzymes are saturated. And this straight line is stationary phase. Stationary phase means, see, Sana, tell me, enzyme combines with substrate and converts them into what?
Um, I'm sorry. Can you repeat that?
Enzymes combines with substrate
Mm-hmm.
and converts them into
The homo.
enzymes combine with substrate and convert them into products, right?
Yeah.
Yes. So here, why we will have stationary? That means the reaction stops. Can you tell me why?
Because
Think. Can you tell me why?
because
Yes. Think. Because
natures.
because all products, all products, sorry, all substrates are converted into products. Okay, all substrates are converted into products. So here, what do we have? We only have free enzymes, but we don't have any substrate to combine with the enzyme. Are you understanding?
Yeah.
Yes. So if I want my reaction to continue, what I will do?
Um,
Yeah, tell me. If I
you
want my reaction to continue, what will I do? See, substrate is not there, only enzyme is there. Now you tell me the answer.
add substrate.
Yes. So I will add substrate so that my reaction continues. Is this point clear?
Yeah,
Yes. Did you understand this? Yes or no?
yeah.
Okay, so this thing is very important. Okay, so Sana, we will do in the next class about pH. We will do about temperature factors that affect enzyme function. Then
Mhm.
we are going to do on water, an important biomolecule. All right, we're going to study on the lipids, and then we are going to start with the nucleic acids. Clear? Have you done nucleic acids?
Yeah,
Yes. DNA and RNA, have you done?
yeah,
Yes. Okay. All right. So today's class, Sana, have you understood?
yeah. I have.
Any doubt?
No.
No. Are you understanding the class? Yes.
Yeah, I am.
Yes. Okay. Fine. So if you have any doubt, you can let me know. Okay.
Okay.
All right. So shall we end the session here?
Yeah.
Okay. All right then. Bye. Take care.
Bye.
Bye.